Well, I hate this. This is our last meeting. I'm going to miss you all. I've really enjoyed this term. And I thought today we'd go over something which is kind of a fun decision that affects you all, especially all of you who are part of the MIT community. I talked about it during class, but it really sort of summarizes everything that we've been talking about this term, and hopefully it will be the product of some discussion here today. And I hope it'll illustrate some of the things, or reinforce some of the things I've tried to illustrate this term for us. But and it is the CRISPR decision. This is actually a very nice diagram of something that bacteria have been doing for millions of years. Does anybody know what CRISPR is? Material science students, not a lot of biology people in here, but so CRISPR has been called the greatest biotechnological development of all time. It stands for clustered, regularly interspaced, short palindromic repeats. Everybody's heard of DNA, right? We all have DNA. All of our cells have DNA from our moms. We have DNA from our dads. That DNA sequence, it's kind of like a deck of cards that have been shuffled. So you don't have usually the exact sequence that you get from one parent or the other. You kind of get the two together, and like a deck of cards that has been expertly shuffled, you have your own unique DNA. And the way DNA is sequenced, you can come up with all kinds of variations, from how tall you're going to be, to what color eyes you're going to have, the color of your hair, whether it's curly or straight, or whether you have any at all. Everything that makes us up as human beings is really based upon the sequencing of our DNA. It is the sequencing of this DNA that also makes us more susceptible to certain illnesses and diseases, and it has promise for avoiding disease and curing diseases. If we could resequence our DNA, if you could imagine your DNA, and you could identify a sequence in it which is responsible for a disease like leukemia, and you could isolate that sequence and with a pair of scissors snip it out and substitute something that that shuts off that predisposition, or that DNA sequence which is responsible for your being susceptible to an illness like leukemia, then you could eliminate. Think of the things we could eliminate. We could eliminate susceptibility to cancer. We could eliminate susceptibility to blood disorders like leukemia. We could also customize babies. You want a baby with blonde hair and blue eyes? Well, if you can do the DNA sequencing, you can literally insert a sequence of DNA that turns off blue eyes and turns on brown eyes, or vice versa. You want somebody with blonde hair, or with a high IQ, or with a susceptibility, or who would not be susceptible to certain diseases. If you can identify the genetic sequence that's responsible for that and substitute it, then you can do almost anything. That's why CRISPR, this gene editing technology, was heralded as perhaps the greatest biotechnological invention of all time. And it happened right here. It's happening right here today at the Broad Institute in Kendall Square. I mean, not more than 100 yards from where we're meeting here today. And it's happening in laboratories all over the world. It's happening at the University of California. And in fact, that's part of what the CRISPR decision was all about. in order to understand the CRISPR decision and its importance in intellectual property law, it's important to understand what it's based on. CRISPR is really a naturally occurring phenomenon. It is something that bacteria have been doing in response to viruses for millions and millions and millions of years. It's a form of developed immunity, which was adapted by genome editing. A virus would enter the organism, it would change the DNA sequencing, and the organism would develop over a period of time a resistance to that, or an immunity to that particular virus. So like many phenomenon or many methods that we have in science, the analog for it has been demonstrated by nature for, as I say, in bacteria's case, for millions and millions and millions of years. And scientists have been studying these things with the hope of learning things that would make all of us better off. This is actually the prokaryotic antiviral defense mechanism diagram. Does anybody know what prokaryotic refers to? Anybody here of prokaryotic? In order to understand the CRISPR decision, if you know, there are two words you need to understand: prokaryotic and eukaryotic. Prokaryotic refers to single-cell organisms, really simple organisms with no definable nuclei. Eukaryotic refers to what? All of us. Complex multi-cell organisms, including plants, animals, and human beings. So this phenomenon, of course, has been something that has been practiced by prokaryotic organisms for millions of years. And somebody once came along and had the idea of what if we could apply this phenomenon, or this method of gene sequencing that bacteria have been using for millions of years. And it's done with an enzyme or a protein which literally snips out the offending DNA sequence and or quiets it or silences it in favor of an immune response. So a fundamental question is that I have for all of you. Since this CRISPR technology is based upon a naturally occurring phenomenon, can you patent a naturally occurring phenomenon? Can you patent naturally occurring substances or not? We've kind of covered this. We've talked about it. What do you think? Can Einstein patent E equals mc squared? I saw somebody shake their head. Of course not. Can Newton patent the law of gravity? Of course not. Why not? Because they're naturally occurring phenomena, right? So, can we patent a gene editing sequence that is based upon a naturally occurring phenomenon? Let's get back to the fundamental definition of what can be invented. This comes right out of the United States Patent Act. Whoever invents or discovers any new or useful process, machine, manufacture, or composition of matter, or any new and useful improvement thereon may obtain a patent therefore, subject to the conditions and requirements of this title. The key words are invents or discovers and new, okay? If you don't invent it or discover it, and if it isn't new, and if it isn't, of course, a useful process, machine, manufacture, or composition of matter, then you can't patent it. So the question is whether or not this CRISPR Cas9 gene editing sequence meets that definition. invents or discovers requires that the patentee be the actual creator and not just the first person to come along, like Isaac Newton or Albert Einstein. But you have to be the actual creator and not something that already exists. Even though discovers makes it seem as though a person can get a patent on some naturally occurring substance, if he or she was the first to find it. But courts have consistently held that one cannot get a patent on laws of nature or natural phenomena. We've covered this, okay? So Einstein's out of luck, Newton's out of luck, okay? Because they discovered naturally occurring phenomenon, which have been around here since the beginning of time. So can you then patent something that bacteria have been practicing right in front of us for millions of years? Well, the short answer is no, but the actual answer is it depends. A natural substance that has never before been isolated or known may be patentable in some instances, but only in its isolated form, since the isolated form has has not naturally occurred, okay? Likewise, something that is an analog for a naturally occurring phenomenon probably qualifies for being patentable. So if the researchers at the University of California, who really were the first to apply for a patent on the CRISPR Cas9 method, were basically creating a tool based upon something that they had observed in nature, but a tool which did not exist naturally, then it's probably patentable. This is a really kind of an interesting question for me because it was never addressed in the CRISPR decision. And I'm going to get ahead of myself a little bit. The CRISPR decision was a big fight over this genetic sequencing technology. The Broad Institute said that we own the patent. University of California said we own the patent. And one of the things that neither party raised in trying to cancel each other's patents was that, well, you can't patent this because it's a naturally occurring phenomenon. Why didn't one of the parties raise that? Exactly. They'd be canceling their own patent. But if you read the CRISPR decision, if you study the CRISPR decision, you know, you kind of wonder whether or not this was a fight that actually could have been had in the first place, because if you read the technology, if you actually read the papers, the first papers that came out that were published in 2012 by the University of California, you kind of sit there and you see these words like prokaryotic, and you think, and you see bacteria. And basically what they're describing is a natural phenomenon in nature that they had essentially isolated in a test tube. What the people at the University of California were able to do is they were able to take this naturally occurring phenomenon that they found in bacteria and mimic it, copy it, and do it in a test tube. And so for that reason, it probably is patentable, but we'll never really know because nobody actually fleshed that issue out when it came to the case, because as we got pointed out over here, if they did, they'd be canceling their own patent, and we don't want to be doing that. So anyhow, it started in 2012 by a researcher by the name of Dudna, who published this paper showing that this DNA sequencing technology can be used in a test tube at targeted sites. And I highlighted that because I think that's what makes this new. Go back to the original definition of what can be patented. Go back to basic principles. I think that's what makes it new, even though all she was doing is describing a phenomenon that has been practiced by bacteria for millions and millions of years. But we'll never know, because nobody ever asked a judge or a court or the patent trial and appeal board to answer that question. But it was her efforts, of course, when she was able to describe this phenomenon in her technical paper, that everybody took notice, because everybody saw the potential for this. There's a potential here for a cure to cancer. There's a potential that actually has been realized for a cure to leukemia. This technology was used in 2017 to cure a child of leukemia. The first time that's ever been done in the history of the world. There are plans to use this to cure AIDS. There are plans to use this to eliminate breast cancer. There are plans to use this to eliminate basically every one of the diseases that results from genetic flaws, or for which we are susceptible genetically. In the same way that you're susceptible to blue eyes, or male pattern baldness, or being 6 foot tall, or 5 foot tall, you have a genetic predisposition to, oftentimes, to diseases. And as scientists and as researchers, we're maybe not so interested in coming up with a cure for being tall, but we are advancing society if we can use this technology to eliminate all illness. And I don't think that it's an exaggeration to say, especially given the fact that this has been successfully applied to leukemia, that this is that it's an exaggeration to say that this is one of the greatest biotechnological revolutions ever to come along. It's kind of interesting, and you can look at the thing. I'll post it on the website. This is kind of cool about how it actually works. But if I could do CRISPR for dummies here, basically what happens is they use an enzyme, a protein, that's the CSA9 substance, which is exactly what bacteria do. They put it in the test tube with the DNA, and it literally snips or cuts out, or actually it silences that DNA, that offending sequence of the DNA, and thereby eliminates. Now, it's a far cry from doing it in a test tube to actually applying it, even to single cell organisms, but also to multicellular organisms. And that's that leap is at the heart of the CRISPR decision. The origin of the CRISPR decision, actually, or CRISPR technology, was first seen years before, in 1987, where a scientist in Japan at Osaka University sort of accidentally identified the CRISPR CSA9 gene editing technology. But the significance of repeating genome sequences was not understood at that time. And so it was a phenomenon that was first noticed back in 1987, but nobody knew the significance of it. It took the University of California researchers to really understand this gene editing phenomenon for it to be significant in terms of a scientific discovery. So a year after the folks at the University of California published their paper and filed an application for a patent, by the way, 2012, so what patent law applied at that time? They filed, the University of California applied for their patent on this gene editing technology in 2012. Was that before or after the adoption of the America Invents Act? Before, exactly. So what kind of a world were we living in the United States when the University of California applied for their patent? First to invent, exactly. And then in 2013, the Broad Institute, by specifically a bioengineer over there by the name of Feng Shang and his team, demonstrated for the first time that they could use this technology on not just on eukaryotes, on multicell organisms. So now they had taken this gene editing sequence and they have been able to develop a technique for using it with multicell animals. So remember, prokaryotic and eukaryotic. Here it is for the first time ever being demonstrated in eukaryotic organisms. And that's significant. So the people of the Broad Institute, not being stupid people, filed for an accelerated review of their patent. And while their patent application was pending, the University of California got wind of it. They filed an interference action, and they asked that the Broad Institute's patent application be denied because it infringed on the University of California's patent application, their discovery. They said, we own this. We've figured out this gene editing sequence, and we filed for a patent application before them. We should own the patent. In 2017, the USPTO, the United States Patent and Trademark Office, ruled that the Broad patents, with claims covering the application for the CRISPR-Cas9 in eukaryotic cells, was distinct from the invention claimed by the University of California. And you already know the reason why. Because there's a difference between eukaryotic cells and prokaryotic cells. The USPTO said that demonstrating this technology in multicell organisms was a significant leap forward from demonstrating it in prokaryotic organisms. Therefore, it made it novel. We've heard that word before. It's not an obvious advance or development from the prokaryotic cell patents. It met the requirements of novelty, non-obviousness, and so the Broad got the patent, and that was the most important part of this decision. And I told you, I think I was sitting there I think I told you I was sitting there in the Federal Circuit Court of Appeals when this case was argued in front of me. I was waiting to argue a trademark case against the same panel of judges that decided this case. And I sat there with all the news reporters and everybody who watched the arguments in this. And the amount of, you'd think that the lawyers that were standing up and arguing this case were the actual researchers who had done the, done the research themselves. The amount of knowledge concerning the biotechnology involved was really phenomenal. And I have to even say that the judges who were deciding this case had to become experts in eukaryotic and prokaryotic organisms and gene sequencing. This is really a very technical area of the law. And it's an area of the law, quite frankly, which is often decided by an understanding of the technology. So the key to the case was the University of California researchers never demonstrated the editing technology in eukaryotic cells. The USPTO ruled it is undisputed that Dudna's 2012 paper does not report the results of experiments using the CRISPR-Cas9 system in eukaryotic cells. And that's true. In fact, I think I mentioned to you earlier this term that in the trial they brought in not only experts in this gene editing technology who talked about, on one side, for the University of California at Berkeley, they said that the Broad's development of this technology was really just a natural development of the work that they had done. Nothing novel here. And then they brought in scientists that testified for the Broad that said, oh, this was this was a giant leap forward. This is the way these cases are decided. And in this case, I think that for the most part, the experts canceled themselves out, except for the cross-examination that took place with the University of California's expert, who had a really hard time when asked about interviews that Dudna had given to the various newspapers when she published her paper about how she had no idea how she could demonstrate this in eukaryotic cells. And so, you know the old saying, loose lips sink ships. I don't know if this is actually the perfect application of that phrase, but I have a sign in my office that says, "No fish ever got caught what kept its mouth shut." And that applies here. If she had kept her mouth shut and not given an interview to the New York Times talking about the limits of her investigative techniques, then her experts never would have been confronted with those words on the stand. She never would have been confronted with those words on the stand. And the USPTO judges who sat in as fact finders of the case would have had a much more difficult time deciding this case, because they wouldn't have known the difference between prokaryotic cells and eukaryotic cells and how this technology applies to them, unless this researcher herself had basically laid down the trail of breadcrumbs for them. Yes?
If she had not given that interview and she had known that she hadn't demonstrated that, but then they asked her, is that fine to be yeah, I think it would also work? Or,
well, yeah, you never know. You never know what would have happened. When you're looking at a case and when you're trying to decide, how to try it, you look for hints. You look for ways of undermining the other side's case. And I'm not saying that the inspiration would not have come separately, but it would have been far less obvious. If some enterprising lawyer had come upon the idea of arguing that there's a difference between eukaryotic and prokaryotic cells, and how the University of California's research had never demonstrated it in eukaryotic cells, that would have had some persuasive effect. But I bet you the experts could have figured out a way of making that not sound as important. When you're paying, $1,000 an hour for an expert to come into court and testify, they have ways of figuring out how to deal with these inconsistencies. But when the researcher goes out and basically, red flags it, it's really hard to overcome. The expert couldn't, and there may have been a valid way of getting around this, but the researcher, with her statement against interest, that's what we lawyers call what she said, almost it constitutes practically an admission, but the expert was left with nothing that they could do. The researcher kind of sunk their own ship, and that's how the case was decided, based strictly on what the researchers said that they could do and what they couldn't do. So anyhow, the University of California argued that the Broad had a reasonable expectation of success, and that they used conventional methods, meaning that the Broad didn't do anything particularly inventive or novel when it came to the development of this technology. And that may have flown. That may have flown if the distinction between eukaryotic and prokaryotic cells had not been, or organisms had not been highlighted by the researcher. This is kind of like where the green tennis balls analogy comes in. Did I mention green tennis balls? In the law, and this is kind of a good thing, this is something that everybody that takes a intellectual property course in law school, if you come away knowing one thing, it's this. It's the green tennis ball example. It's possible in patent law to get a patent on all tennis balls. But it's also possible to get a patent on green tennis balls. We talked about the fan, the ceiling fan, right? I probably should have used the green tennis ball as example. So the Broad Institute has a patent on this technology, this tool that they've developed, okay, for sequencing these DNA using these enzymes that cut out the bad DNA and substitute the good DNA in a test tube. So in a way, you can look at the University of California's patent as a patent on all tennis balls. And the Broad Institute has a patent on green tennis balls or red tennis balls because they've been able to use the technology and apply it to eukaryotic cells. So if you're looking to do some cancer research, by the way, both the University of California and the Broad Institute have turned their research over to universities for free. Remember Tesla and Toyota and BMW and all those companies? They've given away their technology. So people can use it and develop it in any way they want because both patentees believe that the technology needs to be made available generally to universities to do follow-on research. But they still can license the technology to private companies. So let's say you just establish a startup over in Kendall Square, and a few of your really smart biotech classmates have put all their life savings into a new startup that's going to use this gene editing technology to eliminate some disease. What do you do if you're starting this startup company as far as licensing this technology is concerned? Who do you talk to? What you probably have to do is you probably have to license the technology from both, unless you can come up with an entirely new technology, and I'll get into that. So if you're going to get into this, if you're a private company and you're looking to commercially exploit this technology, what you're going to need to do is you're going to probably have to license it from the University of California at Berkeley, and you're going to have to, if you're using it for eukaryotic cells for human beings, you're probably going to have to license it from the Broad as well. And then you can conduct your research. So, as I said, any company that wants to use CRISPR to develop human therapies will probably have to license both technology. The University of California, Berkeley, the reason they lost was, I think as I already said, was that the testimony of their research has demonstrated that the Broad's development of this technology was not an obvious or insignificant advance on the technology, because they themselves said, we can't do it, we don't know how to do it, I don't know if we'll ever be able to do it. And then along comes the Broad and said, we did it. Very hard for the University of California to say, well, that's not novel, when you've just given a statement to every scientist in the world saying that you couldn't do it. As I said, the USPTO cited Doudna's own statements when they found that the Broad Institute had significantly advanced the technology such that its invention was worthy of separate patent protection. By the way, both sides spent $15 million on this legal fight. Remember we talked about the development of graphene? You know, for a long time, and when I say a long time, from 2014 to 2017, which is a long time when it comes to scientific development, for a long time nobody knew who to license this technology from. There was literally a hold on this type of technological research because of the big patent. No one wanted to delve into this because they were afraid that they were going to infringe on somebody's patent. And so while these two sides were fighting it out, spending millions on this, on the right to say that they own the technology, this particular technology was not being used. So what do other researchers do? What do they do in the graphene case? When the Scotch tape method was tied up, what do? Exactly. They found other ways to create these graphene structures. Same thing goes for this technology. What the Broad Institute did is they actually found another enzyme that did the snipping, CFT1 or something like that. So in fact, it was the same group who basically found another. And how do they find it? How do you think they found it? The CAS9 enzyme, that protein was the one that University of California used and which was also exploited by the Broad Institute when they applied it to eukaryotic cells. So the Broad Institute is carrying on their research. They're worried about this patent fight with the University of California, and they're thinking of doing the same thing that scientists did in the graphene case. So what do? How do they find it? Come on, how would you find it? How would you find it? What did the University of California do? I know you know. I know you know. Where would you go? Where do you go to look up an answer to something? Where did the University of California go to look up an answer to this? Yes. Exactly. They started looking at bacteriums again. Bacterium used hundreds of different enzymes to accomplish this same process. So instead of using the CAS9 enzyme, what did the Broad Institute go? They went back, looked under a microscope, watched identified another enzyme that the bacterium was using, and they used that. And guess what they did? Well, what would you do when you isolated it?
Same thing that the bacteria is doing.
Yeah, well, okay, so you've isolated it in the lab. Now you can do it with a new enzyme. What's the first thing you do? Exactly. What are those things called? Patent paper pairs. They wrote a paper and they patented it same time. And guess who owns it? So now you don't have to go to the University of California to use this gene editing technology, because theirs does not cover the new enzyme. It only covers the CAS9 enzyme. So now you go to the Broad Institute and use their alternative enzyme. And that's actually what's happening. Remember what we talked about with the advancement of science, and does patent paper pairs inhibit, does the use of patent protection inhibit the development of science? I say no. I say what it does is it only spurs more human ingenuity. What there appears to be no limit on is human ingenuity. So whether it happens to be a new method for isolating graphene, or gene editing sequence technology based upon different enzymes, different analogs of nature. There is no limit on human ingenuity, at least as far as I can find. And so the only thing that patent protection does is it protects the investment and promotes investment and more science. I can't find any evidence that it has ever inhibited human ingenuity. Anyhow, the USPTO decided in favor of the Broad Institute, and then, of course, what did the parties do? Did they settle the case? Did they agree to a joint ownership of the technology? Nope. They kept spending money on lawyers. Lawyers don't have enough Lamborghinis with IP law license plates on it, so they kept spending money on lawyers. And another $7 to $10 million later, the United States Federal Circuit Court of Appeals, where I was sitting that day, heard the case, and they decided the case. And in 2018, I think September 10th of 2018, the U.S. Court of Appeals for the Federal Circuit decided that the USPTO decision awarding the patent to the Broad Institute was supported in fact, and so the Broad essentially won the case. $20 million later. University of California gets the consolation prize. I think that if you're going to use the CAS9 gene editing technology, you're going to have to license it from both the University of California and the Broad Institute. But if you wait a day, you know, somebody in this room is probably going to come up with another way of skinning the cat. So that's the CRISPR decision in a nutshell. Kind of a fun decision because it really gets back to fundamentals about what is patentable, you know, natural processes, novelty, all those kinds of things. Ingenuity. For me, it's kind of an analog of what we've been trying to talk about here. Any questions? Last day, last chance to ask a lawyer questions and not get charged for it. Once you go out of this courtroom, this classroom, you're going to have to be paying lawyers to answer questions. So anybody have any questions? All right, so no questions. Thanks a lot for coming. I told you that I'd be asking you folks to do a questionnaire. It's quarter of, so I intentionally have left you time. If you wanted to fool around with this thing today, you could, or you could take it home and fill it out. But this is my little questionnaire asking you to sort of evaluate this course and this instructor. And the idea is really not so much that that I learn anything, but what I want to be able to do is I want to design this course in a way that the students that come after you have a better time. I want to know what works, what doesn't work. I want to change the course, improve the course for the students that come after you. So don't think of this as so much an opportunity to talk about just about what sort of experience you've had. But I'd like you to think about the students that will be sitting in your chairs next year and how you would help me improve this course for them by telling me what works, what doesn't work, what you like, what you didn't like, and be as honest and forthright as you can possibly be, because my goal is to just make this course better for the people that follow on. What we presented this term was in part customized by what students before you had to say. So I'd really appreciate your comments. And if you can do it today, that's great. If you want to take it home and think about it and just drop it off with Jerry in Dr. Ega's office, that's fine too. But take a little time and fill it out if you don't mind. It would help, not only help me. As they say, great teachers know that moments of evaluation can and always should become moments of teaching. So it's your turn to teach me something, is what I'm saying. Teach me. And thanks for letting me teach you a little bit about what I really love. Thanks a lot.